Namaskar. Today we'll study anti-HIV drugs or antiretroviral drugs. HIV full form is human immunodeficiency virus. It is a retrovirus, a type of RNA virus. And virus are of two types, DNA virus and RNA virus, because the virus has three components in its cell. The outer envelope containing the lipoprotein envelope, containing the glycoprotein, and the inside envelope there is capsid layer consisting of capsomeres, and the third component is the nucleic acid core, which may be DNA or RNA. And based on that we can have two types of virus, DNA virus or RNA virus. If the nucleic acid component is DNA, then it is a DNA virus. If it is RNA, then it is RNA virus. And one type of RNA virus is retrovirus. In DNA virus, the DNA is converted to messenger RNA. This process is called transcription, where the DNA strand double helical structure is converted to the single strands, and one strand is called the sense strand, the other is antisense strand. The antisense strand undergoes transcription to form messenger RNA. RNA virus directly provides the RNA, but retrovirus, the RNA is first converted to DNA, then from DNA again by transcription RNA will be there. So the first process of RNA to DNA is called the reverse transcription process. So the enzyme there, in addition to RNA, HIV has reverse transcriptase enzyme, HIV has integrase enzyme, which helps in integration of its DNA with host cell DNA, and it has also another enzyme, protease. The glycoproteins surrounded by the envelope are glycoprotein 41 and glycoprotein 120. GP41 is connected to the envelope. To GP41 there is GP120. HIV attacks the T lymphocytes, and the T lymphocytes have CD4 receptor on their surface, and the co-receptor is the chemokine receptor 5, CCR5. And GP120 GP120 binds to the CCR5, the chemokine receptor 5, and GP41 binds to the CD4. And so GP41 interaction occurs with CD4, and GP120 interaction occurs with CCR5, and because of that interaction the virus gets fused or enters into the T lymphocytes. After entering, there will be uncoating, and it will release its RNA because it is a RNA virus, and it will also release reverse transcriptase, integrase and protease. The RNA is converted to viral DNA by reverse transcriptase. The process is reverse transcription. Then this viral DNA will integrate with the host cell DNA to form this provirus, and this provirus will undergo transcription to produce messenger RNA and genomic RNA. Messenger RNA will now bind to the host ribosome to synthesize the viral proteins. Those proteins will be degraded by protease to structural and functional proteins. All the structural and functional proteins and genomic RNA, they will assemble together to produce the new virion, which will be released from the T lymphocytes after bursting of the T lymphocyte. So the drugs used in HIV can be the drugs which inhibit entry or fusion of the virus. Number 2 inhibits reverse transcriptase. Number 3 inhibits protease. Number 4 inhibits integrase. And we can see the classification now. Entry or fusion inhibitors by binding to GP41 subunit and enfuvirtide, by binding to GP120 by inhibiting or by acting as antagonist of CCR5 co-receptor. Example maraviroc, vicriviroc inhibit the reverse transcriptase, which can be either nucleoside analogs or nucleotide analogs or non-nucleosides. Nucleoside reverse transcriptase inhibitors are analog of either thymidine or cytosine or guanosine or adenosine. Thymidine analogs like zidovudine, stabudine. Cytosine analogs like lamivudine, jalcitabine, emtricitabine. Guanosine analogs like abacavir. Adenosine analogs like didanosine. Then nucleotide reverse transcriptase inhibitors. Nucleotide is the nucleoside plus phosphate. So adenosine monophosphate analogs like tenofovir are the examples of nucleotide reverse transcriptase inhibitors. So nucleoside reverse transcriptase inhibitor you can short form say NRTI, and nucleotide reverse transcriptase inhibitor short form you can say NTRTI. Then some reverse transcriptase inhibitors, they directly act at the catalytic site of the reverse transcriptase enzyme. They are not analogs of any nucleosides, so they are called non-nucleoside reverse transcriptase inhibitors, NNRTI. Example efavirenz, nevirapine, delavirdin, rilpivirine, doravirine. Then protease inhibitors like saquinavir, indinavir, nelfinavir, ritonavir, amprenavir, fosamprenavir, lopinavir, atazanavir, tipranavir, darunavir. Then we can have integrase inhibitors like raltegravir, bictegravir, dolutegravir, elvitegravir. And a new drug, bevirimat, is there, which is a maturation inhibitor. It is a semi-synthetic derivative from the Chinese herb Syzygium claviflorum. This is a Chinese herb, and it inhibits maturation. That is, viral maturation involves processing of viral proteins to make virus infectious. And when it is inhibited, there will be release of non-infectious virus which cannot infect the host cells. So these are all anti-retroviral drugs, and they have a tendency to develop resistance. So one term is used called HAART, highly active antiretroviral therapy, in which a combination of antiretroviral drugs are used. So the HAART includes two nucleoside reverse transcriptase inhibitors and one protease inhibitor. So it is a combination of either 3 to 4 drugs, and which may be two nucleoside reverse transcriptase inhibitors and one non-nucleoside reverse transcriptase inhibitor, or one nucleoside reverse transcriptase inhibitor, one nucleotide reverse transcriptase inhibitor, one integrase inhibitor, or two nucleoside reverse transcriptase inhibitor, one non-nucleoside reverse transcriptase inhibitor, and one protease inhibitor. Or it can be one nucleoside reverse transcriptase inhibitor, one non-nucleoside reverse transcriptase inhibitor, and two protease inhibitors. Or it can be one nucleoside reverse transcriptase inhibitor, one non-nucleoside reverse transcriptase inhibitor, and one protease inhibitor. Or it can be one nucleoside reverse transcriptase inhibitor, one nucleotide reverse transcriptase inhibitor, and one non-nucleotide reverse transcriptase inhibitor. So we can see here examples emtricitabine plus tenofovir plus rilpivirine. So this is an example of this one nucleoside reverse transcriptase inhibitor, one nucleotide reverse transcriptase inhibitor, and one non-nucleotide reverse transcriptase inhibitor. Another example, emtricitabine plus tenofovir plus bictegravir. So this is one example of this one. One nucleoside reverse transcriptase inhibitor, one nucleotide reverse transcriptase inhibitor, and one integrase inhibitor. So this is all about antiretroviral drugs. Thank you.